Welcome to the new world, and I am going to take you through a little bit of what we are going to do in biology with what's probably going to be the rest of this school year. I need to keep this shortish, so I'm going to shoot for 10 minutes or less. But here is a brief review of Unit 6. We're going to finish genetics, and then we're going to move on in some way or other. Okay. Whoops, I need to click here. There we go. So remember our unit question, remember from way back ago, our Unit 6 question was, what is the best explanation for the similarities and differences we see in individuals within a species, not only for one species, but for every species of plant and animal? So we went into this unit, the main learning goal from the first lesson was basically just that individual organisms have characteristics that differ from others of the same species. Now, this is intuitive. You've known this since you were a small child. But the variation that we used, that we looked at, were in the coat colors of the jaguars, the insecticide resistance in the mosquito, and then geese that were able to fly at much higher altitudes than other geese. Now, each of these had something that was different about one group than the other, and our goal was to explore what could cause that difference. And here were some of the explanations that we talked about if you didn't already know stuff and you're looking at it like a scientist for the very first time. We have the idea that individuals get their traits from their parents. All right, I can do this now. Right here. I can do that terribly, but okay. The idea that your traits are caused by your genes, which is nice. And then some individuals look different than others because they got a mutation. Spoiler alert, it's kind of all of them, right? You get your genes from your parents, and their expression can be different. Now, you can have a mutation that makes you different from a parent. And we're going to go into a little bit of that going into next week, as into how some of these mutations can arise. All right. Oh, I have to turn off the tool. Okay. Now, lesson 2, the main learning goal there was about characteristics, which were different versions of a trait, or I dropped in that word phenotype, if you remember. The characteristics that organisms exhibit are the result of proteins in their bodies. Proteins perform many jobs in the cells, the majority of the jobs, but not all. The function of a protein depends on its specific structure. If the structure changes, the protein may not function the same way. Now, this was where you put together those little models. We had version 1 where they were all the same, so they all made just a chain. And then version 2 where they fit together in different ways, so they formed different basic shapes. Yeah. This is another thing that we did there when you're looking at different proteins. You were asked to, here we go, talk about the type of protein. And then you were comparing the black jaguars to the spotted jaguars. You know, are these the same? And in this case, hemoglobin, the amount of hemoglobin in the red blood cells in black jaguars and spotted jaguars were about equivalent, both high. The difference we found were in the eumelanin. Here we go. Which is a pigment protein. And in the black jaguars, there were high levels in the fur, and in spotted, there were low levels in the fur. And the other major difference was here with the MC1R, regulatory protein, high levels. Whoops. See, part of the protein deleted. And then the protein was complete. The regulator was complete, the MC1R. And the, here we go. Oh, that takes those away. Okay. All right, so wait, let me finish what I was trying to say. The MC1R, right, so you had high levels, part of the protein was deleted, and the protein was complete in the spotted jaguars, which would lead one to suggest that maybe the normal condition was the protein complete condition, right? That was what you see, what it means. Okay. After that, we went into the protein structure, which was hinted at in lesson 2 there, or said. The structure is determined by its amino acid sequence, and the amino acid sequence is what determines the protein structure, right? And here is just a reminder, you had the, what's this tool? I don't even know. Wait. This is a different color. We had the amino group and the carboxyl group. This was the basic fundamental structure of all the amino acids. The R group was different for every amino acid. And it had this other hydrogen out here. So remember, you cut out the pictures and to make a chain. And so if I go here, oh, interesting. I see. Now, we had these levels of structure. You didn't have to know this, so if this is like, wow, I don't know what this is, that's okay. I just showed it to you on the side there, giving you the idea of how it changes shape. So the primary sequence is just the linear, just the literal sequence. And then those spontaneously will form certain shapes that form more complex shapes. But if we look back here at some of these R groups, you see, these are going to force different conformations, different shapes in the chain of amino acids. So remember how they bound? Dehydration synthesis, the amino group and the carboxyl group. I chucked out a water there. All right. It's a lot different instead of being able to at least get some kind of, recognition of something from some of y'all. But that's okay. I'm at almost 8 minutes now. So lesson 4, main learning goal. Structure of DNA contains information for assembling amino acids into proteins. A segment of DNA nucleotides that codes for a specific protein is called a gene. There are people that will argue that the regulatory elements are also part of the gene, but we're not going to worry about that right now. The coding sequence is what we're going to talk about. Remember, we had the phosphate, the sugar, and the base. We had our cytosine, our thymine, guanine, adenine. Remember, the T goes with the A, the C goes with the G. And it forms hydrogen bonds between them and base pairing. And that code gets turned into RNA, which gets that's transcription. This is translation. First letter, second letter, and then the third letter. So we use the circle one. That's the handout that I gave you. This is just it in table form. But it's the same deal. We have first position here. We have our cytosine and our uracil, second letter. So we're all here. Third position doesn't even matter, does it? It's all going to lead to leucine. All right. So lesson 5, the main goal was to introduce you to the messenger RNA. I showed you the video that showed splicing even, which is, a next level of information that you haven't had yet. And but anyway, the messenger takes the DNA message to the we did the play to the ribosome where it gets translated. And RNA, the differences were on the 2' carbon over here. And so for the sugar, that is the difference here. We have the hydroxyl here. And okay, there. And DNA is arranged in the cell. It's in the nucleus. And coming up, we'll go into chromosomes and whatnot. But now I'm going to make a little brief quiz. I'm at 10 minutes right now. And I'll just put that up, and you can do that at your convenience over the course of this week. Do Sunday night, unless they tell us differently. All right. I hope everybody's okay, and hope you're taking precautions. And I might see you again, but maybe I'll see you on the Google Meet thing since the Zoom bombing happens. So we're not probably going to do Zoom. All right. Anyway, take care.